And we're live. So thank you everyone for joining us today. This is our weekly Friday seminar in the Open Source Neuro series, which is part of the Worldwide Science Initiative. Our series is all about open tools and methods in neuroscience that we collect on our website over at open-neuroscience.com. Don't forget to upload your projects to the website and sign up to give a talk of your own. We welcome contributions and love if you can follow us and spread the word. Today we have the pleasure of hosting Dr. Jason Nasse. Jason is a senior scientist at Addgene, specializing in viral vectors and science communication. He received his PhD in neuroscience from Ohio State University, where he focused on synaptic integration and GPCR-mediated plasticity. Throughout his graduate and postdoctoral training, Jason spent much of his time characterizing and troubleshooting AAV vectors in both the central and peripheral nervous systems. Following his postdoc, he decided to leave the bench behind and took a position at Addgene, providing technical support to scientists using AAV vectors. We have heard about AAV vectors in one of the previous talks in this series, and will now hear in depth from Jason about the Addgene AAV Data Hub. Please feel free to ask questions in the YouTube live chat or join us over in the Zoom room. The details will be posted shortly. So hello, Jason, and thank you for being with us today. Please go ahead.
Thanks, Cecilia. I appreciate it. And hi, everyone. Thank you for coming to this talk. I'm really excited to talk to you a little bit about Addgene and about our AAV Data Hub. So I'm going to break this talk into three different sections. The first, I'm going to give an overview on Addgene and kind of our mission as a nonprofit. And then I'm going to talk to you a little bit more about our AAV viral service and our AAV Data Hub, which is something that we initiated about 1.5 years ago, and we're really interested in expanding it out and sharing it with the rest of the community. And then I'd really like to open it up for discussion. So if anybody has any questions or would like to have a dialogue, we'd really appreciate any feedback that you have from the community itself. So a lot of people know, and a lot of people don't know, surprisingly, that Addgene is a nonprofit. We started as a nonprofit plasmid repository back in 2004. And the premise of this was that one of our founders, Melina Fan, was having trouble getting quality reagents. And she thought, there must be a better way than just talking to different labs and trying to get plasmids from individual investigators. So she started Addgene as a repository for individual plasmids. And so, to introduce this, how Addgene can be really important, I kind of want to give you a little bit of a story about my own history with Addgene and with reagents. So when I was in grad school, and this is about 10 years ago now, so I'm dating myself a little bit, general-rhodopsins were really starting to become hot, and everybody wanted general-rhodopsins, but there wasn't a lot of options as to what type of promoters you could get or anything else. And so there's a specific group of neurons that I wanted to transfect and activate with general-rhodopsins, but they were mixed within a bunch of different phenotypes, and there wasn't a specific vector available to do this. So I decided that I was going to do this. So I read through the literature. I found a really small promoter that would fit within an AAV vector. I got the general-rhodopsin vectors, and then I went looking through Addgene to see if I could find anything. Luckily, they had both plasmids that I needed. And now, as Cecilia said, I was trained as a neurophysiologist. So molecular cloning was a bit of a mystery to me. And this first picture here is an actual slide that I presented after I did a bunch of my cloning and strategy, because I was so proud of being able to learn how to do restriction sites and ligations. And I presented it, and everybody who was a molecular biologist looked at me and said, that's really easy. You don't really need to say that. So I got a real reality check. But I took this information, these two plasmids, and I made my vector. And then I spent the next couple of years getting viruses made and characterizing it and making sure that it worked. And I was really excited about it. And then my time at graduate school was ending. And I said, okay, I should probably share this because it's good for the community. And so, after I did this, I made the plasmid itself, I went to a collaborator's lab and I did what everybody does. I put it in a freezer and it looked much like this. And I thought, okay, it's there. I'll come get it when I need to. So I was deciding to go deposit this. I went over to the freezer and said, all right, well, let me get my materials and take a look at them. And what I actually saw was this. This is almost exactly what I saw. The freezer that had my plasmid in it was wide open. It was completely empty. There was nothing in it whatsoever. And I thought, well, they must have moved it. So I talked to our collaborator and they said, oh yeah, that freezer thawed and we threw everything away. So my 2 years of work are now completely gone. And this is a really good lesson for somebody who spends a lot of work in doing something, because Addgene's mission is to make sure that this doesn't happen. And so the way that Addgene is set up, we have a workflow which goes something like this. A researcher makes a plasmid or modifies a plasmid. They send it to Addgene. Addgene takes this plasmid. We make files of it. We perform QC to make sure that the sequence is there, make sure everything is intact. If there's any discrepancies, we go back to the depositor and say, hey, this is a discrepancy. What do you think about it? And then we start to distribute it. When we do our QC and make sure that everything's validated, we also make archives of that plasmid. So this plasmid is now not in just one freezer, but it's in 3 freezers. So if anything happens to any one freezer along the way, we have backups, which means your plasmid is now preserved, it's quality controlled, and we can help you distribute it. So like I said, Addgene's a nonprofit. And the reason that we're a nonprofit is because it helps us make decisions that are going to help scientists. We don't have to worry about making money. We don't have to worry about investors. We can focus on doing what's best for the scientists and what's best for the scientific community. It also lets us have a lot of fun. We call ourselves Addgenes, and everybody's friendly. We're collaborative, and we really focus on doing the best that we can to help support the scientific community as a whole. So we're here to help you be successful in every way that's possible from the resources that we have. And that's a really great mission to be a part of. I love it. So sharing with the community has a lot of benefits. So if you're an individual lab and you make a plasmid and say it's a wonderful tool, everybody loves it and they want it. If you're a small lab, you now have to, if somebody requests this, you have to spend the time and effort to package it, to make sure it gets to the place safely, to make sure that there's any technology transfer agreements put in place. And then somebody in your lab, probably a grad student or a research tech or a postdoc, is going to have to do this for you because you're the PI, you don't have time for that. Addgene takes all of that out of the lab and does it for you. It's free to deposit with us. We have a global shipping network, so we can now ship, we've already shipped to more than 160 different countries. We have, we've tested all of our shipping materials. So when we send viral vectors, we send viral vectors around the world. And we have things set up. So if we're sending from the United States to Australia, we have systems in place to make sure dry ice gets refilled over there and make sure that everything gets intact and gets sent and remains in pristine condition. It helps you stay organized. You no longer have to keep track of which box all of your plasmids are in or where the sequences are stored on some graduate student or postdoc's computer. We can keep all that information for you. So we're not just a plasmid repository, we're also a knowledge repository, which allows us to provide a great resource to the scientific community. I'm going to talk about this in a second, but when you deposit with Addgene, it's been shown that you get increased citations for your papers because more people are using your plasmids because they're more readily available, and then that helps get feedback back to you. It also is beneficial to you as a reward system. If people order your plasmid, we will give you reward points, which then you can use for discounts on your own plasmids. So sharing with the community and doing it through Addgene has lots and lots of benefits. So I talked about increasing citations. This is some data that we looked at a few years back, and we analyzed a large number of papers. And the important part to notice here is that when you deposit with Addgene, which are the papers that are in the blue lines, you get much more citations than you do when you don't deposit with Addgene. And that can be reflected also in the second graph, as in we're showing not just increased numbers, but if you have a more recent paper, there hasn't been enough time for citations. But if you look at the fold increase over papers that do and do not cite Addgene or do not contain Addgene deposits, there's almost a 7-fold increase in the number of citations you get. And that's good for you as a scientist because citations and increased impact is what helps you to get grants and what helps you to increase your ability to actually do the research that you want to do. Sharing your resources also helps to rapidly distribute new and important materials. So 18 months ago, COVID started to happen around the world. And Addgene, excuse me, Addgene put forth a concerted effort to get COVID plasmids and try to make them as easy to get as we can. And so since March of last year, we now have over 4,000 plasmids that we make available to researchers. Researchers then can use these to develop things like vaccines, like testing and therapeutics, so that we can understand how COVID is spreading, how COVID is interacting with the body, and we can do it in a really rapid way because we can distribute things really fast. We typically send things out within 2 to 3 days, and they get to their responders the next day. So being able to share everything in one central place is really great for the entire research community because it makes things easy to get back and forth amongst different labs. So we have a number of different materials and everything is centralized. Like I said, we do distribute things worldwide. We also have a bunch of data. So all of the sequences for all the plasmids are found on our website. You can download those and you can look at each individual sequence. We sequence every plasmid that we get with NGS sequencing, so you know exactly what those sequences are. We do guaranteed quality control. We control for all of our viral vectors, which is something I'll get into in a little bit more detail later in the talk. We control all of our plasmids, and if there's a problem with something that you find, let us know and we're going to make it right. We're going to contact the depositor and figure out what we can do to make sure that you have all the information that you can. Protocol sharing. So we have a wide variety of protocols on our website. Our main website, we're getting almost, still talking about impact, we're getting almost 2.5 million page views per month, and almost greater than 200,000 of these are new users. So there's a great exposure at Addgene, which if you deposit with us, then that exposure then gets magnified because all of your deposits are here and your information is here so people can readily access it. In addition to plasmids, though, we also have a wide range of educational materials. So on our homepage, if you click on the Education and Tools section, we have a blog post, which gets almost 120,000 page views per month. On this blog post, we cover subjects like CRISPR, we cover lentiviruses, we cover AAV viruses, molecular tools, new things that come up in the community from all range, from all different disciplines, because we have a number of different disciplines that we look at. We have a number of protocols. So we have protocols for lentivirus. We have protocols for producing adeno-associated viruses. We have cloning protocols. So the resources that we have available for the scientific community is another great resource that we try to provide as a place that does free sharing and data sharing. We have ebooks, so we have details about how to do plasmids and CRISPR and viral vectors. We even talk about science careers. A lot of academic labs, as new scientists are graduating, going through grad school, there's a wide range of careers that aren't talked about or that aren't known. So Addgene has spent a lot of time, trying to discuss some of these different careers that you can have, and let young scientists know how to get into those, which I think is an excellent resource. And we also have a really great YouTube channel. You can go to our YouTube channel. You can see videos on making plasmids, on doing PCR and doing quantitative PCR. It's a wonderful resource. I invite anyone to come and check out our YouTube channel. We also have a really robust help center. So we hire PhD scientists who are experts in plasmids. And in my case, I handle the viral vector side of these. So if you have questions on your AAVs, if you have questions on plasmids, you can send us an email and we will help you in the best way that we can. It's a really great resource. We get thousands of requests every year, and it's a great pleasure for us to help our customers as best as possible. So the collections that we have at Addgene has been growing from the start. Just this month, we've reached over 100,000 different plasmids that are now contained within our repository, which is an amazing milestone. We're really excited about that. We have plasmid kits. We have pooled libraries that you can use for CRISPR screenings. We have AAV vectors. Again, I'm going to talk about this in a little bit more detail a little bit later in the talk. We have a number of lentiviral vectors that we can use. And we announced towards the end of last month, we're starting a new initiative, and this is in collaboration with the neighbor project at UC Davis, where we're going to be starting to produce neuroscience-based or recombinant antibodies. It's an exciting venture that we're working with the Jim Trimmer lab at UC Davis, and he's been in charge of the NeuroMab project. If there's anybody out there who's heard of that, I think it's a great resource. It's been around for a number of years now. And we're going to collaborate with them to try to produce and distribute more neuroscience-based recombinant antibodies. We're really excited about that. So I'm going to focus a little bit now on Addgene's AAV Data Hub, or AAV viral service. We launched this viral service in late 2016 as a way to produce high-quality, ready-to-use AAV vectors. Since a relatively modest start in 2016, I think we had around 40 vectors. We now have over 600 ready-to-use viral vectors available. We work in collaboration with the UPenn Vector Core, which used to be one of the main distributors of AAV vectors and producers of AAV vectors. So we took on their catalog and act as a distributor through that site. And currently we have over 10 different serotypes. We have 20 different promoters. We've put out we've distributed almost 45,000 units of AAV to scientists around the world. And this averages to about almost 1,000 a month, which is a huge number that gets sent out. And we guarantee shipping, we guarantee the quality. And I think it's a really great resource for researchers around the world. So with AAVs, they're tricky to use. If anybody's ever used an AAV vector, you read a paper and you say, oh, well, everything's great. I'll put it in my cells and everything works great. One of the things that's really important to that is the quality of the reagents that you have. Addgene produces AAV vectors in a relatively large volume, so we can produce fairly big lots that can ensure consistency across your experiments. We do titers with digital PCR, so we get a very accurate titer for each vector that we do. And we also sequence every single lot that we put out. So we take the viral vector after it's been packaged, we extract the DNA, we perform next-gen sequencing on it, and then we make sure that the sequences match up exactly to the plasmid vector, so that when we send out a viral prep, you know that you're getting the exact gene packaged into that was contained in the plasmid as the original. We also perform sterility tests. So in culture, without any antibiotics, we add the viral vectors to different cell lines, depending on what the promoters are, making sure that there's no bacterial or fungal contamination. We do in vitro screenings. Since a lot of our viral vectors do have fluorescent proteins, we can see that there's expression with those viral vectors. So we can say that the viral vector is clean, it has the genes that were in the original plasmid, and that it's expressing in the cell types. So when you get a viral vector from Addgene, you can have confidence that it is of high quality and that it's exactly what was deposited with us, which I also think is a really great resource because viruses, a lot of people are making viruses in the lab and they're really difficult to produce to make sure that they're pure enough, and also to make in high enough quantities. So we try to take care of that, and we do it in a way that we try to reduce the cost as much as possible. As a nonprofit, that allows us a little bit of flexibility in being able to produce things as close to cost as we can. So when I said, in terms of quality, with that plasma that I had packaged back in graduate school, when I had that package, I had there was a lab that was helping us as a collaborator, and we gave them our sequence, and they said, okay, we're going to package it into a virus and we'll get it back to you. And they did, and I injected it, and this is what I saw. Basically, it was kind of a necrosis, a necrotic AAV, and I have no idea why. So we contacted them, and they said, well, we don't know. It should work. Having a repository or a place that produces things and produces the amount of the high quality and has a number of checks and balances that we have is really beneficial to the community, because we guarantee that what we say is in our package, what we say is in the vector is in the vector. And that saves researchers time and effort, because they're not going to have to go through something like this that I did after 2 months and figure out, all right, well, is it my vector? Is it because my titer's wrong? And you don't have any information from it. So I think that we do a really good job to make sure that we give consistent, high quality vectors, which helps the community as a whole. Now, like I said, I handle the vector, the scientific support for Addgene vectors. And I think as of this morning, I looked over the past year, I've had over 800 different questions from scientists that are using our vectors. And so I've gotten to get a little bit of idea of like the types of questions and the types of issues that a lot of people are having when using AAV vectors. Some of the things that we get a lot of questions are Cre-dependent vectors. So the vectors that have LOXP sites, and they're only supposed to express in Cre. Sometimes there's leaky expression with these, which is a known phenomenon. I'll get into in a little bit more detail in a couple of slides. People have a lot of questions about serotypes. Each serotype can transfect different cell types in the brain. They have questions about using dual viruses at the same time. How is my retrograde virus working? Should I dilute my virus, and what should I dilute my virus in? These are all really, all these factors are things that can make or break your experiments, and they require a lot of troubleshooting. They require a lot of finite manipulations to make sure that you're getting the right injection volumes, the right injection titers. That's what the AAV Data Hub is here to help provide information for. So Addgene launched the Data Hub in 2019, and it's a completely open platform. Anybody can look at the data, and anybody who's using a virus can share, an Addgene virus can share their data. We do accept negative data. A lot of times the information that you need about a virus isn't what does work, but it's all the things that didn't work. And sometimes getting that information is really difficult, because people don't publish all of the things that they tried before the thing that worked. They publish what worked. And that may be different if each individual brain's region and each individual neuronal or cell type, if you're looking at astroglia or microglia. So this is some data that I generated when I was trying to determine serotypes. What I did here is I injected 4 different serotypes. It all has the chicken beta actin promoter and expresses GFP. It's all in the same brain region. It's all done with the same age rats. And it shows kind of the diversity of serotype, of cell types, and diversity of expression patterns you can get with the same genetic construct, but with different serotypes. We know that Addgene has 10 different serotypes. We know that there's over 180 identified brain regions. There's hundreds of neuron phenotypes as determined by, there's a really big effort right now to determine genetically differences in neurons. And there's also species differences. So there's a lot of variables that can occur with every single AAV experiment that you might want to set up. So in this experiment that I did, we used AAVs 6, 8, 1, and 9. And what we found was that you can see that the green is the GFP, and then I was trying to express into neurons that express norepinephrine. So what's in magenta here is dopamine beta hydroxylase immunostaining. And what you can see is that even though, each of these injections shows great expression across the whole region, the number of double labeled neurons varies dramatically. So in AAV6, even though it's getting good expression in these motor neurons, it's getting almost nothing in the dopamine beta hydroxylase positive neurons. With AAV1, it was a little bit better, still great expression in neurons all the way around, but only 23% double labeled. AAV8 and 9, both great, 82 and 84% expression, dual expression with the DBH neurons. So this is just one example that can show like how understanding and knowing what AAVs work in a given brain region and with a given cell type can make a huge difference. And what we really want to do is try to harness as much experience from the community as we can and get that data into the AAV Data Hub so that people can spend less time troubleshooting and less time working hard on the part of the experiment that doesn't generate the important results. Because it can take months and months for each individual injection in order for you to get the results back. Another question that we get a lot of information, another aspect we get a lot of questions about are Cre and flip dependent vectors. This is one of our vectors that we have data on from the depositor, and it shows great results. It shows that in a wild type, you're getting not much expression whatsoever. You're getting good Cre dependent expression. And this is a lab that has spent a lot of time honing the exact injection parameters that work. When you're doing injections with flex vectors, it's known that they recombine. And there's a paper that came out a couple of years ago that's showing that with these vectors, there's a small percentage that gets recombined just by amplifying the DNA in bacteria before even packaging into AAV virus. And these are different from each lot to lot. When Addgene does our quality control with the next-gen sequencing, we're able to quantitate the exact level of recombination for every lot of vector that we put out. And then we can give you that information so that you can it can help you to guide how to titrate out your injection parameters. It's a really important aspect that I think people who use these vectors, the flex vectors, they have learned about it, but people who are new still have trouble with it. And so if we can get more people to deposit the data about each individual lots or each individual serotypes or each individual type of construct, whether it be a DREAD or generorhodopsin or GFP, then it can help minimize, again, how much time people spend troubleshooting and optimizing just the injection parameters themselves. So the mission of the AAV Data Hub is, just as I've been saying, is to get all this information in one central place and to get as much details as we can to help scientists around the world. And so we take both negative and positive data. In this particular figure here on the right, these are two different species. This is the same lab, two different species. And they found with a retrograde virus, and they found that they get really good transfection in cortical neurons that they know project to a specific region, but they weren't getting it in other regions that they know. Negatively, they have the same thing. So this is a hamster, this is a mouse. Again, they inject, this time it's a retrograde virus, but it's Cre expressing, so it should only get into these chat Cre mice. And they're seeing the same phenomenon in terms of the retrograde vector not getting into neurons that are in regions that are not the cortex. And so this is a really interesting phenomenon. And it's something that, when we read papers on retrograde viruses and things like that, it's perceived that, put it in there and it works great. And in some regions of the brain, it does. In some regions of the brain, it doesn't. And that's one of these caveats that I think we can really try to work on trying to get more information about. And if you consider kind of the things that we talked about before, whether using a rat or a mouse, or if you're in a non-human primate, or if you're working in, we have data that's submitted from zebrafinch. All these species in different brain regions can have a lot of different properties. And so we really want to try to get as much information as we can to work these out. So one of the things that I like to think about is like, what makes an impactful submission to the Data Hub? What are the things that are going to help benefit the community? And I want to put this out there so that, if people are watching and they think, yes, I have data, yes, I want to submit it. We want to try to guide people so that we can provide as much information, as much useful information to the community as we can. So this is some of the things that we think about when we're looking at how to structure a deposit of data. When you're making an injection, there's a lot of people use a lot of different techniques now to inject. Some people will use a Hamilton syringe. Some people use a pulled glass pipette. Some people use a nano injector. And the size of those can make a huge difference. Whether you pre-wet them with PBS before drying up your virus can make a big difference. Do you coat your glass with SealGuard, or do you coat your glass with other type of siliconized agent to prevent the virus from sticking as you're injecting it? Do you pull a long pipette? Do you pull a short pipette? All of these can make really big differences in whether the virus gets localized in one small area, whether it gets up the injection tract. And so one of the things that we'd like people to consider is give all that information. This particular deposit was made in the Data Hub, and it shows, sorry, my mouse is a little bit finicky. It shows the injection site, so you can see how much the virus spread. It shows the transport into the fibers, so you can see how well the expressed protein is getting transported into different regions, into different structures, whether it be dendrites or axons. And then he provides really great information on the injection parameter itself. So he says, 0.8 microliters is perfect for the medial septum. It doesn't go too far lateral or ventral. He gives the timing that he left the injector in place. He gives the rate of injection, whether it's in like a single bolus or it's done over multiple boluses. So all of these types of information are things that we think are really important. Another important thing to consider when you're submitting a Data Hub entry is what are the images that you're submitting? What do they actually show? I really like this particular figure because it goes through a lot of different parameters. In this case, he's injecting a retrograde virus into one area of the brain, and then he's injecting a flex virus or DIO virus into another region of the brain. So the Cree will get taken up at the terminals, transported back to the cell bodies where he injects a different virus and then gets expression. So what I love about this is that one, he's giving a good diagram of where he's injecting. He's giving a nice low mag of the injection site, so you can see what expressed there. He's also showing the transported region. So up in this is the prelimbic cortex. He's showing the cells that got theoretically taken up with the Cree, and then now they're expressing the dread virus. Then he shows functional images. So here he's got a dread virus. He gives CNO, and he sees an increase in expression of FOS. So he's given a functional output. He's also given some behavioral output by showing he's showing the FOS expression by quantitating across the cell type. So he's showing injection site, he's giving immuno for a functional output, and he's quantitating the functional output. So this gives somebody else all the information they need to try to replicate this type of experiment in their own lab. And I think that's really important. So again, when you're considering what types of data to include with your images, the things that are important are what cell types are you trying to get into? If it was a specific cell type, do you do immunohistochemistry for the cellular markers for that? And does it co-localize really well with the virus that you're expressing? Did you quantitate any of that? Can you put that into a figure that you can look at and see? If something didn't work, did you try more than one virus? Do you have those images? These can be multiple submissions, and we recognize that it takes a lot of time to do this, but I think the benefit to the community and the benefit to people as a whole as they get more and more data into this can be really be impactful on research. Again, this is negative or unexpected results. We recently put out a survey to our customers that are using AAV vectors regarding our Data Hub. And the number one thing that they said that would help improve what we have in there is negative or unexpected results. I think it was over 76% of the people who responded said we would love to see more negative or unexpected results. And this really drives home how difficult it is sometimes to get these AAV experiments to really work. And if anybody out there has actually done any of these experiments, I would love for them to think about how much time it takes to do these and how much time it might take to actually make a data submission and how much time you could save if somebody had done that for you prior to doing that. I think it's a really great way to think about what's the benefit of depositing data and what's the outcome of it. In this case, again, retrograde transfection didn't happen in one brain region, it happened in another brain region. This particular data submitter, he's using a GCamp, which is a calcium indicator, and he sees expression of calcium indicator, but he's not getting any dynamic changes in this particular cell type. So this might not be the correct construct for this, or it might not be the right serotype, but this is the type of data information you can say, okay, I worked in this particular area of the brain. Maybe this construct doesn't have enough dynamic range. Maybe there's too much intrinsic activity. I can try a different one and try to hone things out. And if we get more data from the different types of these biosensors, then we can help people to kind of select which one may work best in their cell type and in their brain region. So again, negative data. A lot of people are afraid of submitting negative data because it's kind of looked at as, as not being productive. Maybe you did something wrong. Maybe there's something wrong with, your parameters. But if you've done the proper controls and you're still getting the exact same results, then it may not necessarily be that you're doing anything wrong. It just may be one of the properties of the virus itself or with the promoters that you're using. And that's really important information that I don't think gets disseminated quite enough. And so we'd really like to encourage people to, if you're working with a virus and you're doing everything well and you're controlling for things and it's not working, submit that data. Let other people know. Let them know what's not working for you. And then as a community, we can come together and make things better. Details of expression. Again, this comes down to doing your due diligence with serotypes and with the types of neurons you're trying to get into. So when you submit data, did it get into your cell types? Did it not get into your cell types? Did it get into cell types that were around yours, but not into the one that you're particularly interested in? So in this particular case, this person was trying to get into the SCN and states, AAV8 didn't get in there very well, but AAV1 did. So that's great information that tells people, all right, this works, this doesn't work. It shows expression. And then people can use that when they're trying to order viruses or when they're trying to design their own experiments. So one of the things that's a little bit more difficult, but is really important we have a number of new fluorescent sensors. There's dopamine sensors, there's norepinephrine sensors, there's acetylcholine sensors. And the way that these operate may be really different in between different brain regions that express these transmitters. And understanding like how the dynamic ranges of these may work in different cell types is also something that's really important. And so if something doesn't work in your system, like with the previous example with the GCamps, that's great information to let people know. And you may not be able to get that into a full publication, but you can deposit it in a place like Addgene's Data Hub, and you can show what's working and not working. And then other people can learn from that and try to design their experiments in a little bit more detail in a little bit better way. So why share your AAV data? I think I hope I've made a pretty good case about that at this point in time. But basically, AAV experiments are not straightforward. They may seem that way from publications, but they're difficult. They take time. It can take months or longer to optimize your experiment. Say you use one serotype, it doesn't work. Use another serotype, it doesn't work. Now you're at 4 to 6 months and you're still not getting it to work. So deposit that data, save somebody else that time, and then hopefully they can deposit and save you some time with your next experiments. Sharing helps speed up research. I think, as the example I gave with the COVID resources, that was a rapid distribution of them. And just think about how much time that saved just by getting those reagents out there and getting the information out there. And more importantly, we want you to go from having this type of experiment where something might not work to, having this with your experiments. It's going to save you time, it's going to save you effort, and it's going to save your lab money, which is really, really important. One of the things that we found from the survey that we put out is that a lot of our customers didn't know that they actually can submit data to the Data Hub. So I want to let you know you can. If you have an AAV vector from Addgene that you're using, you can submit that data to the Data Hub. And we encourage you to do that. Right now, all you have to do is go to our website. It's listed here, and I think it's also in a link. And you can just click on this contribute data, and you can submit it. If you have questions, you can email us. We're happy to answer questions. We're happy to help you submit the data in any way that we can. We're currently working to improve the Data Hub. Right now it's in a beta version, and we're working really hard to get feedback from the community to make it an even better resource. And to wrap things up in terms of like the full talk that I'm giving, we have a number of resources that are here to help you. If you're using our vectors, or even if you have just general questions, you can send us an email at the Help Center at help@addgene.org. We have a number of protocols that I mentioned before, how to produce AAV vectors, how to titrate them, how to do quality control on them. We have a great blog. We've got almost 25 posts on how to use AAV vectors and different properties of the AAV vectors. And we recently started this new initiative. It's called Addgene AAV Office Hours. What this is it's a way for you to schedule a time to talk with us about your experiment. A lot of times in email is really difficult to try to get all the information that you may need from us or the information to us. So these are designed to have one-on-one conversations. You can share your data on screen with me, and it's completely confidential. We won't tell anybody about what we see in your data or anything like that. It's really just there to try to help you optimize your experiments and provide as much guidance as we possibly can to do this. This has been in effect. We launched a pilot with this starting in January. We've already had a number of people participate. We've contacted people now in over 9 countries, which I think is phenomenal. It's really exciting, and we'd really love for anybody who's interested to sign up, have an appointment. We'd love to help you. So what I'd like to do now is, if anybody has any questions, I'd really like to kind of open things up for a discussion. If you have any questions, or if you have any comments, if you have any information that you want to provide to us, we would love to hear from you. Like I said, we're in the process right now of trying to improve our Data Hub. We want to revamp it. We want to get more data into it. We want to get more data that the community needs into it. And so we're soliciting feedback so that we can do that in the best way possible based on your own needs.
So thank you so much. That was amazing. Really, really nice talk, very in depth. Thank you for sharing your story and the philosophy behind it, right? I think it's very revealing of the problems that are very common in science. Surely can resonate with many people. So I'm checking to see if we have questions over at YouTube, giving people some time. Maybe someone here in the Zoom room would like to ask a question. Just pop it in the chat or unmute yourselves. Jason said he would help getting your experiment running. I'll go ahead. It seems to be silent for a time being, but so does the deposit work with a DOI or how, in terms of citing that? Is that can you explain how that works, please?
So that is something that we are actually we're looking at addressing right now. One of the questions that we did ask the community is what is the interest in having a DOI, and there's a huge interest in it, and it's one of the things that we are discussing, and that's part of the revamping of the Data Hub right now. It's something that we do want to provide, and we're trying to look at how what's the best way to provide the most information for people, and that they will get the most benefit for providing a DOI and being able to be cited from that.
Yeah, that definitely makes sense. And is there an ongoing survey that people can? You mentioned surveys along the way. There's something open now that they can answer questions about, or is it more feedback in an
so
email?
Right now, so we sent this survey out to a number of customers. We've closed it since then. But we would welcome any feedback. You can send me an email at I have my email is put out there. You can send me an email. You can also send it to the help desk if you have questions or comments in the Data Hub. You can just put attention Jason on it, or AAV Data Hub on there, and we'd be happy to answer your questions there and get feedback from that.
Great. So we'll be looking forward to people submitting your questions over there, since there don't seem to be any at the time being. Maybe I was wondering if you'd like to comment. You were saying about working with scientists around the world. What can we learn from that, or maybe any future initiatives in this, spreading and the education aspect that you mentioned that Addgene is working on. Like, is there anything upcoming that you'd like to comment on?
So yes, we're going to be doing soon. I'll be doing an official launch. Like I said, I did a soft launch on the AAV office hours, where I was mainly soliciting through questions we get from our help desk. But because of the success of this, we would like to expand it a little bit more. And so we'll be putting together a blog post and talking a little bit more about that. So keep a lookout for that. Also, we are working on more content for the AAV blog. So in questions like with the flex vectors and stuff, we're going to be working on some content for the blogs so that we can increase the amount of information we're providing on those and help scientists, better strategize how to perform those experiments and how to, test each lot. The important thing with the flex vectors is that, every single lot you get is going to have a different recombination rate, and they're all going to have to be titrated to make sure that you're getting specificity. And it's daunting, but it does have to be done with every lot and every vector that gets put out.
Yeah, of course. So hopefully we'll be on the lookout for that, and everyone can contribute. So I also like one last comment is that it was so interesting that you mentioned the incentives, right, to share the work. Sometimes that is very hard to do, and here you're pushing for negative data as well. So with the kind of benefiting points and the increase in citations, right? Mm-hmm. So hopefully more and more people contribute to this research, and they have this talk to go back to. Really, thank you so much for going into detail about all of this. Thank you.
Yeah, thanks everyone. I appreciate it. This was a lot of fun.